Thank you, Bruce, for the opportunity to come talk about some of the work that we've been doing focused on histone methyltransferases as potential therapeutic targets in leukemia. And Jonathan's talk was a perfect introduction for what it is that I'm going to talk about. However, I'll be talking a little bit more on the development and gene expression side of things as opposed to the cell cycle side, but maybe we should be thinking about that in more detail. And as Bruce and Jonathan have both mentioned, I think many of us, and obviously I'm biased because I'm working on this, but think that inhibition of histone methyltransferases, demethylases, DNA methyltransferases has significant potential in terms of development of anti-tumor or new therapeutics, much like the kinases that you heard this morning. But we're still very early in our developing our understanding of what these modifications and enzymes are responsible for. So as Jonathan mentioned, one of the histone methyltransferases that's actually been known to be mutated in about 10% of human AMLs or human leukemias is the MLL gene located on chromosome 11q23. So this gene was cloned about 20 years ago now, and it was only about 10 years ago that it was shown to be a histone methyltransferase. And just recently, we're starting to understand what it is that the translocation actually does in terms of driving the development of the leukemia. So this gene is consistently rearranged in infant acute leukemias. This is how I became interested in it. I'm a pediatric oncologist about 10% of the time. And if you find this translocation in a leukemia in an infant, it predicts a very poor prognosis, which is unlike most of the cases of pediatric leukemia. As you may know, those are generally fairly curable diseases. It's also rearranged in about 10% of adult leukemias and also in many leukemias that develop after treatment with other types of chemotherapy, such as DNA-damaging agents such as topoisomerase II inhibitors. And the wild-type function of MLL, or one of the wild-type functions of MLL, is to maintain homeotic or HOX gene expression during development. So this was shown in the mid-90s through knockout studies in mice. It wasn't clear at the time how it was doing this, and then subsequent studies demonstrated that this C-terminal set domain, this is the histone H3 lysine 4 methyltransferase domain. So this is an enzymatic portion of the protein. You see the protein is actually quite large, and when the translocation occurs, invariably you have the amino terminus of MLL fused to the C terminus in frame always of the translocation partner. And there are some 60 different translocations of MLL now defined. However, 3 or 4 make up the majority of the translocations. The one I'll talk most about today is the 911. This is the most common translocation, most often found in acute myelogenous leukemia. So you'll immediately note that when the translocation occurs, this enzymatic portion of the protein is gone and something else is replaced. So my lab's been studying MLL rearranged leukemias now for about 10 years, and we initially defined the FLT3 receptor tyrosine kinase as actually frequently mutated in these leukemias, and we decided that the field would move on in terms of kinase as much as we've heard this morning, and went to focus on what it is that the MLL-Fusion protein is doing to induce leukemia. We've recently shown, much as you've heard from others, that the Wnt beta-catenin pathway is important in the development of this leukemia. But we figured that we really have to know molecularly what's going on here if we want to develop a true targeted therapy. And I would argue, at least in leukemia, the only targeted therapies that have made a significant difference for patients are those like Gleevec, which target directly the action of the fusion oncoprotein BCR-ABL, and all-trans-retinoic acid, which again directly targets the fusion protein, in that case, RAR-PML. So I don't have to give you much background in terms of the modifications. You just heard about these from Jonathan. Just to reiterate that the H3K4 trimethylation that MLL normally places is found near the transcriptional start sites and is believed to be somehow involved in allowing polymerase II to bind and initiate transcription, but just this modification doesn't appear to be enough. And then when polymerase II elongates throughout the body of the gene, you see the appearance of these modifications, H3K79 dimethylation and H3K36 trimethylation. And I say you see the appearance because it's still not entirely clear what these modifications do. It's just known that they're associated with transcription. So over the past 3 years, and the reason we got particularly interested in the 3 or 4 years, the reason that we got particularly interested in this is a number of complexes have been purified, all of which somehow influence the activity, or at least associated with active polymerase II. And a number of the proteins in these what's now known to be 1, perhaps 2, and maybe this is all one big complex, it's not clear. But there are 14 proteins here associated with elongating polymerase II, and 10 of them are MLL-Fusion translocation partners. So that tells all of these, everything in yellow here is an MLL translocation fusion partner. So that told us that something about these complexes are critical for the development of leukemia, and given that this is an enzyme that at the time not much was known about, we thought we would start to study HIT.1L, the only known histone H3 lysine 79 methyltransferase, as a potential therapeutic target in this disease. So the hypothesis at the time and still today is that when the MLL translocation occurs, you replace the C terminus of MLL with a protein that, at least in some cases, is directly bound to DOT1, and you therefore get mistargeting of DOT1 to MLL target genes. And that somehow then leads to enhanced expression of the MLL-Fusion target genes, and therefore perhaps inhibition of this enzyme might be a therapeutic opportunity in this disease. So the first thing that we did, again, a few years ago, is did ChIP-Seq analysis to look genome-wide to see if there was any abnormality in H3K79 dimethylation in, this is 3 MLL rearranged ALLs compared to normal developing human pro-B cells shown here. And this shows the presence of H3K79 dimethylation across the HOX loci between HOXA5 and A10. This is important because MLL fusions at the time were known to bind to this region of the genome. And if you look more broadly at sites where the MLL fusion was known to be bound, there appeared to be a significant amount of H3K79 dimethylation. But in this study, we couldn't directly connect the target genes to an aberrant amount of H3K79 dimethylation. So we turned to a little bit more tractable system that was human leukemias. This is now going to be mouse studies where we can introduce the, in this case, MLL-AF9 fusion oncoprotein either into mouse stem cells or mouse progenitor cells. And we've characterized in great detail the leukemias that arise in this model. And we know that the leukemia stem cells, and you heard cancer stem cells alluded to this morning, this is the cells that are the most potent at inducing leukemia in secondary, tertiary recipient mice. We know exactly what the immunophenotype of those cells are, and the frequency of initiating activity is somewhere on the order of 1 in 6. So this is a very highly purified population of leukemia stem cells and allows us to do a number of types of experiments like gene expression profiling and ChIP-Seq analysis that we just recently published a few months ago. So we isolated an HSC-enriched population, not pure hematopoietic stem cells, normal developing myeloid progenitors, and leukemia stem cells. And in the leukemia stem cells, defined the MLL-AF9 target genes, defined all of these modifications. I'm just going to talk about H3K79 dimethyl, but we published all of this data recently. And so then to look at the profiles of this modification in the various normal cell types and the leukemias. So this is just a genome-wide view of where H3K79 methylation is found in the MLL-AF9 leukemia stem cells. This is a representation of the mouse genome. And each place there's a yellow slash is a place of enhanced H3K79 methylation over background. And the first thing to point out is there's a lot of H3K79 dimethylation across the genome. And in fact, the majority of highly expressed genes are decorated with H3K79 dimethylation. So you should immediately start to worry about the specificity of any type of therapy that targets a modification that's found associated with essentially all highly expressed genes. But that didn't deter us. We decided to go ahead and look further. And also the MLL-AF9 is bound to approximately 120 targets, you know, depending on where you draw the line in the statistical p-value, but somewhere on the order of 120 to 130 targets. The vast majority of genes that have this modification are not MLL-AF9 targets. Then if you look at the analysis of the summed scores of all of the H3K79 dimethylation across the genome, now this is looking relative to the transcriptional start site. You heard that H3K4 trimethylation usually has a peak right here near the transcriptional start site of a gene. This is this is our data, but previously published data had demonstrated that H3K79 dimethylation peaks just downstream of the transcriptional start site and then slowly decreases over the body of the gene. And that's exactly what we saw in our leukemia stem cells, and that the most highly expressed genes are most associated with the modification, and as the gene expression decreases, so does the presence of the modifications. This is exactly what had been published before, but what we really wanted to know was what did we see when we looked at the MLL-F9 target genes? Could we find anything aberrant in terms of the amount of H3K79 dimethyl? And this is what is shown here. So this is now the profile of the H3K79 dimethyl around the MLL-F9 target genes, and then these are just other highly expressed genes, either in red and green. So you can see that what appears to be happening here is the MLL-F9 is bound. There's more H3K79 dimethyl than you would expect based on other highly expressed genes, and it's actually spread in both directions. So there does appear to be an abnormality here at the direct MLL-F9 targets. And then if you look at other modifications, H3K4 trimethyl, which is a mark associated with active gene expression, H3K27 trimethyl, a mark associated with repression, and H3K36 trimethyl, there were no abnormalities there. So this seemed to be very specific, this abnormal profile for H3K79. That was mouse. This is human. Don't need to pay attention to this. This is just the HOXA loci again. But here's that summed score for the MLL-F9 targets as defined in the mouse, but now looking at human leukemias. And you can see that this elevated H3K79 dimethylation at the MLL-F9 targets is also found in human leukemia. So this appeared to suggest that, in fact, there was something abnormal going on here in terms of the amount of this modification where the fusion oncoprotein is bound. But it obviously doesn't tell you that the leukemias are dependent on this enzyme. So the next step now, it was to make a genetically engineered mouse where we conditionally inactivate DOT1 and see what that does to the leukemia, and also see what it does to general transcription. At the time when we were making the mouse, we were still quite worried that these modifications that are found everywhere are going to lead to complete transcriptional collapse. Looking back, that's a little bit naive, I guess, to have thought that, but that's certainly what we were worried about at the time. So we just made a model where we could inactivate the gene by putting recombination sites on either side of exon 5, which has the active site of the or encodes for the active site of the enzyme, and then when we express Cre-recombinase, that's taken out, and the enzyme is inactive. In fact, it's not expressed. And here's the type of experiment you can do now once you have this model. Introduce MLL-F9 into mouse bone marrow cells from mice of various genotypes, and in this case, we introduced the two known targets of MLL-F9, HOXA9 and MEIS1, that in combination are known to transform mouse bone marrow, but shouldn't, perhaps, if MLL-F9 is using DOT1, this shouldn't depend on DOT1 unless transcription was just globally affected. So this perhaps could have been a rescue or a control. We then inject those mice or cells into mice. 80 days later, all of the mice develop leukemia. We then sort out the leukemia cells. They're marked with GFP. And then we can introduce Cre-recombinase to take out DOT1. So this is a question, after the leukemias develop, if you take out DOT1 via introduction of Cre, what happens? We can confirm the genotype. We can look to make sure the H3K79 dimethylation is gone. We did that, and it was. We can put them into tissue culture. We can inject them back into mice. So this is what happens when we put them in tissue culture. This is 10 days for a control leukemia, 10 days after plating in methylcellulose. These colonies are prototypical blast colonies that arise when leukemias are cultured in this way. This is, you can see an immature phenotype of those cells. These look like leukemic blast cells. And this is H3K79 dimethylation, just showing that indeed it's present in the wild type cells. And here's what the cell colonies look like after DOT1 had been inactivated. The cells had a morphology of much more differentiated cells, and the H3K79 dimethylation is gone. So this shows you the inactivation of DOT1, at least via this genetic mechanism, leads to differentiation and perhaps decreased growth of the cells in vitro. So if we injected those cells soon after inactivation of DOT1 via expression of Cre-recombinase, injected them into mice, here are the mice that received the cells where DOT1 had been inactivated, and none of those mice came down with leukemia. So this is a little bit unfair. We're inactivating the DOT1 before we inject the cells. We've gone back now and done this where we can, in another way where we can inactivate DOT1 after the leukemia has already developed, and we similarly see a significant prolongation in survival. In that case, we get cells that grow out that are DOT1 not deleted, again showing very tremendous pressure to keep this enzyme for the leukemia to develop. But I haven't shown you anything yet in terms of specificity. So this could potentially happen if you inactivated DOT1 in any proliferating cell type. And this was the first evidence that perhaps there could be some specificity in terms of inhibiting DOT1 in leukemia. So here we've transformed the mouse cells with HOXA9 and MEIS1 to DNA binding proteins known to transform mouse cells that also happen to be MLL-F9 targets. We inactivate DOT1. You can see this is the H3K79 dimethyl blot. That's all. The H3K79 dimethylation is gone. And this is the colony morphology in the absence of DOT1 at the end of 10 days. So I think you can see at least the morphology of the colonies, and if we count the colonies, there's really no effect. So the colony forming activity at least 10 days later is unaffected by the absence of DOT1, whereas in the presence of MLL-F9, these colonies were small, diffuse, and the cells were differentiated. So here's the first evidence that something different is going on in the MLL-F9 leukemias than goes on more broadly. I'll have to say before we get to it that we still don't know exactly what K79 dimethyl does normally. These cells may give us the opportunity to start to look into that. So what about gene expression? As Jonathan alluded to, these marks are at least associated with gene expression. Whether or not they're involved in the control of gene expression, all of them, is still a bit up in the air, but we decided to go ahead and take a look at gene expression in much the same way that I just showed you, where we induce a leukemia, then introduce CRE or a control, and then sort out the cells where DOT1's gone 3, 5, and 7 days later, and do microarrays and say what's changed. Again, we were expecting to see pretty dramatic changes in gene expression. However, after this is now 5 days, at which point the DOT1 is completely gone, only 72, again dependent on how you do the statistics, genes had decreased in expression, about 60 had gone up. And if you look at who the genes are that are decreasing in expression, this is highly enriched for the MLL-F9 target genes. So here's a connection between the oncogene, the genes that are bound, or the genes that are targets of the oncogene, and decreased expression after we inactivate DOT1, suggesting again that there's something about these genes that are more dependent on the presence of that enzyme than genes more globally. And this is still a short-term experiment, meaning 5 to 7 days. We're looking at longer time points now to see what happens as the cells are cultured. And I'll point out that we've also been able to isolate clones, HOXA9 MEIS1 transform clones that have now been growing in the cells in culture for 3 months, so in the absence of DOT1. So it still doesn't, DOT1 is not absolutely required for all gene expression and proliferation. So I'll skip that and end in the last few minutes here with a little bit of data from a recently developed small molecule that was developed by our collaborators at Epizyme. So they've been focused on developing specific inhibitors of various methyltransferases, DOT1 being one of them, and we recently published this small molecule, which is an S-adenosylmethionine-like molecule, a competitive inhibitor of S-adenosylmethionine, that you can see here. This is a Western blot in 2 MLL rearranged cell lines, is fairly potent at inhibiting H3K79 dimethylation in a 411 and a 911 rearranged line, and relatively specific, at least as assessed by these various modifications here for H3K79 dimethylation. They've done extended biochemistry also to look to see the relative specificity of this small molecule, and it seems to be fairly specific. And the possible reason for that is DOT1 is not structurally very similar to other lysine histone methyltransferases. It doesn't have a set domain. Its enzymatic active site looks more like an arginine methyltransferase, but it's not perfectly like that either. So there might be something unique about the active site of this enzyme that allows one to develop these relatively specific inhibitors. So if you look at gene expression, after you treat cell lines with this being an MLL-4, MLL-9 cell line with increasing concentrations of the inhibitor, the target genes, HOXA9 and MEIS1, as I've been talking about, their expression level goes down, as you would expect. And then if you look more broadly at a signature found in human MLL rearranged leukemias, the treated cells seem to have suppressed that signature. And if you look at our MLL-9 targets, those genes also decrease in their expression when you inhibit DOT1 in these human leukemia cell lines. So this shows that a small molecule, at least in terms of gene expression, has very much the same effect that the genetic inactivation did in the mouse model. And this was the first evidence in a human system that inhibition of DOT1 could have some specific anti-leukemia activity. So here are two MLL rearranged cell lines. This is growth of the cells in the absence of the inhibitor, and then the growth of the cells in the presence of the inhibitor. And two things to point out, or here, one, it appears that we are getting inhibition of proliferation and increase in apoptosis, which I'm not showing you here. But a very important thing to think about in terms of further development of any epigenetic therapy is you'll note that these cells grow just fine for up to 7 or 8 days, even though we know by about 3 days the modification is gone. So it appears that the effects of these type, at least this inhibitor and perhaps others, are relatively slow in comparison to kinase inhibitors or other things that directly inhibit apoptosis. And here's a control non-MLL rearranged line where H3K79 dimethyl is inhibited or repressed, and that has no effect on the proliferation of jerkets. And this is just an extension into more both MLL rearranged and non-MLL rearranged cell lines, and this specificity for inhibition of proliferation and induction of apoptosis remains to be MLL specific. So at least in the setting of leukemias and leukemia cell lines, inhibition of DOT1 seems to have a relatively specific anti-leukemia effect. I will say, and some of you are probably wondering, what is the role then of DOT1, and does it have an effect on normal cells? And we've inactivated in normal hematopoiesis, and the hematopoietic stem cells do seem to be affected, and there are fewer, but it takes months in order for that to develop. So we still don't know what it's doing. It's certainly playing some important role, but at least in terms of the time, it appears that this is happening more quickly than the effects on normal hematopoiesis. And in the studies that have been done with this small molecule in mice, even though it doesn't have tremendous PK, there doesn't seem to be toxicity, but that will be borne out over time. So this is just the last slide showing in the mouse system now, MLL-9, HOXA9 MEIS1 transformed cells, just as we've shown for the genetically inactivated DOT1. If you inhibit DOT1 with the inhibitor, you still see this specificity between the two different leukemia lines. So I'll just summarize that various MLL fusions promote enhanced H3K79 methylation. Suppression of DOT1 either by genetic inactivation or a small molecule leads to differentiation and death of both human cells and mouse leukemias. And the gene expression changes are most pronounced for the MLL target genes at day 5. We're hopeful that this is telling us that there really will be a therapeutic window for this, but we've still got some work to do to understand it. And then small molecule inhibitors selectively kill MLL rearranged leukemia cells. So many people have been involved in this. Catherine Berndt is the fellow who drove the development of the knockout mouse. Amit Sinha is our computational biologist. Nanju did all the ChIP sequencing experiments that I've shown you. Here are our collaborators at Epizyme and our long-standing collaborator, Andrew Kung, at the Dana-Farber. So thank you for your attention.
So I guess a simple-minded view is here you have the MLL fusions and HOXA9 is being activated, and that's the critical event. There's various ways to activate HOXA9, some with the MLL fusions, other with other chromatin fusions, such as the NUP98 PhD finger, and then presumably you could tell me about some yet altogether different ways of activating HOXA9.
Well, there are a number of different types of translocations. NPM mutations, which are frequently found in AML, also lead to a signature where there's elevated HOX gene expression. So about 40% of human AMLs will have high-level HOXA cluster.
And the response to the knockdown of DOT1L is totally specific to the ones that recruit MLL, is that right?
Well, we can't answer that yet because there's not a particularly good cell line model for those types of leukemias that you're talking about, and we're just getting all the various mouse models in to be able to address that. So it's a good question. Will this remain only a target for MLL rearranged leukemias, or all those others that express HOX genes? And we just don't
there
know.
was one cell line on your graph which showed some inhibition, which was not an MLL rearranged cell line. I don't know if you
Yeah, that was, what cell line was that?
that one right there.
Yeah,
Just skip it.
that.
That
So
guy.
yeah, this is an interesting cell line that it's a TEL AML1 rearranged ALL, which generally doesn't have HOX gene expression. However, we've profiled that line a long time ago, and for some reason, over the 30 years that line's been in culture, it now expresses high levels of HOXA cluster genes. So what does that mean in terms of real biology? It's hard to know.
So Scott,
Jonathan.
question. So have you looked to see what happens to 79 trimethylation? And then the other question I'd ask in conjunction with that is, in the yeast model, right, 79 di and tri seem to have a differential effect as marking G1S type genes and so on. Do you see any, when you're looking at this, do you see any of that recapitulated in the human or mouse models?
It's a good question. So in Ali Shaliotoford's group and a couple of others have shown that in the yeast, inactivation of dimethyl has more of a cell cycle effect than inactivation of trimethyl. I'm not exactly clear what the effect is there. The antibodies to H3K79 trimethyl for mammalian cells are just still substandard, so we can't address that question. And it's a very rare modification in mammalian cells. I don't know about in yeast, so it's just hard to see. In terms of dimethyl, what we don't know yet, and I think based on your work and others, is in the absence of DOT1 in non-MLL lines, do we see a cell cycle effect? And I will tell you, they grow a little bit more slowly. If you have a competition, the non-deleted cells will always win, but it's not so dramatic that you can see it without paying close attention.
Scott, any explanation for the very slow turnover of the mark and the very gradual effects of inhibition on cell behavior and gene expression?
So the slow turnover of the mark's a little easier than the slow response in terms of gene expression. So this is the only known modification, and there's no known, not that people haven't looked, demethylase. So presumably it just takes proliferation some time in order for this mark to dilute over time. But still, there's another few day delay before the genes change their expression. So that suggests that perhaps the chromatin has to remodel or something else has to happen in order for the repression or suppression of gene expression to happen. And we're starting to look into that, but don't know the answer.
So what does this say in terms of therapy? Would you have to continue the treatment for a long time to see effects?
Yeah, I think germane to the discussion that was being had today in terms of do you need to inhibit things in the enzyme to 90% and consistently, and I'm afraid in this case it's going to take significant and continued inhibition in order to get the effects that we can do in vitro, but we'll see how that plays out in vivo.
Okay, other questions for the speaker? Can't see, is there one back there? Yes.
Since you have a DOT1 condition knockout mice, have you looked at its role in hematopoiesis?
Look in terms of erythropoiesis?
Yeah, use some MX1 CRE.
Yeah, so we've crossed it to VAV, and we've also crossed it to MX. And in the VAV-CRE mice, which are born, so that in that case, DOT1's inactivated in all hematopoietic cells relatively early in development. The mice are actually born at Mendelian frequencies, which surprises us a little bit, but they are fairly profoundly anemic. And this would go along with some of the other studies that have been done using constitutive knockouts that suggest perhaps more of an effect in erythropoiesis, as you're suggesting, than myelopoiesis. So yes, we are seeing an effect on erythroid development. We haven't gone into detail to know exactly what stage of development is affected, and we'll get there at some point.